Hello, everyone. My name is Daniel Mobilio, and today I'll be presenting my summer research that I've conducted under a research scholarship that I received from the Brain Tumour Foundation of Canada. So my project was to develop a migration assay, basically trying to model and characterize how cancer cells or the capacity of cancer cells could migrate. And so the importance of this is because our lab focuses on brain metastases. So if you don't know what brain metastasis is, it's when cancer goes to the brain or spreads to the brain from a primary cancer. So in this slide, we see how breast cancer is able to leave its primary tumor, get into the circulation as you see over here, and then it travels through the blood system and finds its way into the brain. And it does, and basically the cells go through a lot of different changes as they go through this process, which is called the metastatic cascade. However, for my project, we were interested in the migration of these cells, as I said before. And so the simple question we asked was, can we model this in vitro? Or that basically means, can we model the migration of these cells in a dish? And so to do that, I had to develop this migration assay that you've seen in this first slide, and I'll get into more details later. However, at the start, I thought it was probably most important if I just talked about some facts of brain metastases. So brain metastasis is actually the leading form of brain cancer, and it's surpassed primary brain cancers, and primary brain cancers basically means that the cancer just arisen in the brain and only in the brain. And so a secondary tumor is what a brain metastasis is, and that's when it goes to the brain. And so a few studies have shown that there are certain primary tumors that go to the brain preferentially, and the three different primary cancers that has been shown to definitely specifically go to the brain more than any others is melanoma, which is skin cancer, and then you have breast cancer and lung cancer. And so these three different primary sites of origin, they constitute 80% of brain metastasis malignancies, which is a very high number. And so through my studies, we basically determined the migration capacity of these three different primary tumors. And I'll get into more of the details of what different cell lines we use and all of that. But before we go on to that, I also wanted to mention that brain metastasis therapies right now are very slim to none, and the average lifespan of a patient after diagnosis of brain metastasis is around 4 months. So it's a very quick disease that has very bad bad outcomes for patients. And so along with developing this migration assay, we want to use different drugs that work against brain metastasis. And we've shown in publications in our lab before how these drugs could be used against brain metastasis. And we want to show that and see how these drugs work against the migration of cancer cells to the brain, because maybe this could be a way of maybe preventing brain metastasis. So to get into more of the research side, so the main objective, or the main limitation and the hardest part of this whole assay or protocol was to optimize the cell count for for basically modeling the migration of these cells. And so if you had too few number of cells, you would see something like it's like something like this on the left-hand side of this picture, where there's not that many cells in the plate, and it doesn't really give a good readout of migration. And if you plated too many cells, then it ends up being the same thing, where you don't really see a good migration of the cells, and all the cells end up being everywhere. And so it takes a long time to actually do the experiments to optimize the cell count for this assay. And the experiment itself takes 1 to 2 weeks. And something important to note is that the cell count, for whoever would want to do this assay, depends on the cell line being used. And we have to keep in mind that there are different media conditions that people use for different cell lines, and that could affect the readout of the study, as because some components of media could increase the proliferation of cells. And so you may be reading out the proliferation of your cell line in question, other than the migration, which is what we really do want to see. Um, okay, so cell count was a very important thing. And so once we were able to optimize all the cell concentrations and densities for all different cell lines, we were able to move on to next step, which was to actually conduct the migration assay. And so if you see in the left side of the slide here, it's the same picture I had on the first slide. And this is basically an overview of what the migration assay entails. So probably the most important part of this step is that we use these silicone inserts that are able to be placed into a 48 well plate. And so these silicone inserts, they have this special by well structure, where you could put cells into each well on the left and right, as you see in this image. And when you put these cells into these two wells, um, there's a divide, there's a wall, and it allows for the cells to not be able to move into this space here, which is just labeled with empty space from cell migration. Um, and so this part of the assay takes around 2 days. And so the first day just entails you plating the cells and putting the insert on. And the first day of this whole assay is basically to allow the cells to grow into these two separate chambers and allow them to form a monolayer of cells. And this is important because what I've said before with the cell count optimization part of the whole protocol, you need to find the right amount of cells to put into these areas, where after 1 day, you'll have a monolayer of cells, and you won't have like too few amount of cells, or you won't have too many. And so after the first day, and you end up getting this nice monolayer of cells, you're able to take off the silicone insert and place this plate into an imaging system that we use called the IncuSite, and is able to capture multiple pictures at any time point that you tell the machine to do so. And so then you're able to get these cool videos of the cells migrating into the wound. When I say wound, that just means the empty area of where the cells were inhibited from going in the first day of the assay. So this IncuSite is able to generate these cool photos and videos, which is good because it generates a qualitative way of understanding the migration capacity of cells. However, we know that that's not good enough, and we know that we can't just qualitatively say something is true. So the last part of this protocol is to quantitatively determine the migration of cells, that I'll talk about later. So the next slide here is just talking about the IncuSite, which I talked about briefly, but I want to highlight that it takes 3 to 7 days for these cells to actually migrate into the wound. And it definitely depends on the cell line that you're using, but the cell lines that I used, it generally took 3 to 7 days, just depending on the cell line. And so I just have some representative pictures of what this actually looks like from the InQ site. So the InQ site is able to take 9 pictures of the same well. And so what you see here is basically 2 different cell colonies that I was talking about before is these 2 different colonies are the monolayer that I would inject into the 2 biowells of the silicon structure. And then this is just an image of a time point for one of my assays. And the green that you see here is just a dye that I use called Cytox green, and it's able to only go into dead cells. And we use this dye just to understand more of what's actually happening to these cells. And it wasn't really for any other purposes. And it was just a good way of seeing like that the cells that are migrating are actually alive. And so um, this is a picture that was taken at a later time. This wasn't at time 0 or anything, because at time 0, these 2 colonies would be like a monolayer, as you can see on the right side. This is definitely not a monolayer. This is a lot of cancer cells all on top of each other, and they're all too compact. So this was a later part of the assay. And I do have another picture here. And you could see there's a lot more cells in this one. And this is just a reminder that this was not at time 0. This was at the I think at the end point of the assay. So you could see, at least with the InQ site, that the cells are definitely proliferating. However, they don't actually migrate into this wound here, which is what we're most interested in. And so the last was just data analysis that took roughly like 1 to 2 hours. And basically what this data analysis entails is you would calculate the area at time 0 of the wound, and at every time point, you would calculate the area of the wound. And so what you would see is in a control well, meaning that a well that didn't have a drug put into it, you would see that it would probably it would migrate faster than a well that was had a administered drug put into it. And so you would see that quantitatively that you're able to show that the wound or the empty space that the cells were prohibited in going into, um, that space was covered a lot faster with a control well versus a drug. And like I said before, we just calculated this through image J, where you're able to take a picture and outline the section of the well where there was no cells. And then you would compare that to time 0, and you're able to get the percent wound coverage of the cells, which is analogous to the migration capacity of these cells. Okay, so all of this, all of this stuff that I talked about was for this project, which was determining the effect of novel small molecule drugs against the migration of brain metastasis initiating cells. And so this is just a poster that I presented at SNO, which is the Society of Neuro-Oncology, and they hosted a brain metastasis conference in Toronto in late August of this year. And so what this poster kind of shows is how drugs that we found to work on brain metastasis in vivo and in vitro, meaning that the drugs were able to significantly affect brain metastasis tumors in animals and in a dish, were able to affect the migration capacity as well. So on the left-hand side of this poster is just like the introduction to brain metastasis and what I was talking about before at the start of the presentations, and and the objectives was just to determine how these drugs affect the migration of these cells. And so as you can see in the middle of the poster is the data that I produced. And so BT478 is a lung to brain metastasis cell line. and we use drug A. Unfortunately, I'm not allowed to disclose any information about these drugs for intellectual property reasons. However, we've shown through this assay that drug A was able to um, basically prevent not prevent exactly, but definitely slow down is a better way of saying, better term to say, of slowing the migration of these cells. And the bottom of these graphs, they have DMSO, IC50, and IC80. So DMSO is just a control. IC50 is the concentration of the drug that kills 50% of the cells, and IC80 is this concentration of the drug that kills 80% of the cells. And this actually reminds me that another reason why we use cytoxicrine, which labels only dead cells, is we wanted to show that the drugs are not the drugs that we're using are not just killing the cells, and that's why they're not migrating, but actually inhibiting the migration. Um, and so analog on the right side of both of these figures here, analog is just a variation of drug A that we have made with collaborators. And the reason why we made this analog is because drug A is not able to penetrate the blood-brain barrier very well, which everyone has. And the blood-brain barrier is basically um, a very a very dense, compact amount of blood vessels that protects your brain against infections and sort of those sorts of things. Um, However, the problem that we face for brain cancer and targeting brain cancer is that drugs are not able to actually pass the blood-brain barrier all that well. And so it just adds another complication to studying this disease. So therefore, since drug A we know worked well in vivo and in vitro for brain metastasis, but we know it didn't cross the blood-brain barrier that well, we've created an analog or a different variation of drug A that is able to cross the blood-brain barrier a lot better. And these graphs here just show that they both work similarly in inhibiting or preventing slash slowing down the migration of the brain metastasis initiating cells. And I didn't talk about brain metastasis initiating cells, but it's a very important thing to touch upon. And so a long time ago in our lab, we've kind of discovered this cell population that we named brain metastasis initiating cells. And we've defined this cell population as as the cells that initiate um, a brain metastasis malignancy. And there's papers published from our lab that show that this is true. And in all of these experiments that I've done, I've done these experiments with brain metastasis initiating cells. Um, and so since we hypothesized that these cells initiate secondary brain tumors or a brain metastasis, we thought it would be important to model the migration of these cells, since these are the cell or the culprits that lead to brain metastasis. And so what I talked about before with the BT478, which are lung to brain metastasis, the BT673s that we have are melanoma to brain metastasis. And as you can see in these graphs here, drug A and its analog both worked equally well as the lung to brain metastasis in slowing down the migration of these cells. And so basically, we were able to conclude from these studies that this migration assay was able to model migration, which was the first objective and was the hardest to do. And the second conclusion is that these drugs that we've discovered in our lab are able to significantly slow down the migration of these cells. And one last thing I want to highlight from all of this research is that this migration assay is not could not only just be used in the context of cancer, although obviously we're interested in cancer and brain cancer. This migration assay could be utilized for any sort of study which wishes to determine the migration of any cells, and it would and determining the migration of cells is important for different fields of research other than cancer, such as immunology, embryonic development, and those sorts of things. So it's important to keep in mind that although this migration assay was very good in characterizing the migration of cancer cells, it could be used for other applications as well. And finally, the last thing I want to highlight, I cannot zoom in on here, however, just like there's a few representative images on the bottom left of this poster that show how at day zero, it's very small, but at day zero, there's um, there's a very clear separation of the two colonies that I've been talking about in the assay. And at day two, some migration into the wound, and at day four, there's a lot of migration into the wound. Um, and basically, we were able to show and analyze this empty area to create these graphs. And right here is um, it's just a bigger image of time zero of the migration assay. Okay, so I would like to thank everyone that has helped me with this project. I would like to thank the Foundation, Taite Boomer Foundation, for allowing me to pursue brain cancer research in the lab of Dr. Sheila Singh. And I'm very grateful to have that opportunity. Um, and yeah, so thank you very much for listening. I hope you you were I hope that this presentation was somewhat interesting and cool to listen to. And also, if you have any questions about the research and current research that's going on, you could email me here with this email that I have at the bottom right of the slide, or you could call or text me. I don't mind with this phone number, or you could contact Susan at the Brain Tumour Foundation of Canada, and we could go from there. Thank you very much.